Greetings, we are from small group discussion B7, and the topic we are going to talk about is the role of gene promoters in the transcription process. My name is Kenisha Tedijanto, and my last three-digit student number is 154. A gene is a length of DNA that codes for a specific protein that will express a specific feature. By the process of transcription, the template DNA strand will be converted into mRNA and serves as the beginning of protein synthesis in cells. Gene expression is important because it decides how often will the gene be transcribed to produce a desired concentration of the protein product. This is where gene promoter, together with other factors, exists to play a role.
My name is Kinan Kalisha with the last three digits of my student ID number are 152. So the problem formulation of the student project are 1. What is the mechanism of transcription? 2. What is the characteristic of a gene promoter? 3. What is the relationship between gene promoter and the transcription process? And 4. What is the relationship between promoter genes in transcription mechanism and certain medical conditions?
My name is Nesa with last three digits student number 156. The objectives from the student project are: first, knowing the mechanism of transcription; second, knowing the characteristic of gene promoter; third, learning the relationship between gene promoter and transcription process; fourth, learning the relationship between promoter genes in transcription mechanism and certain medical conditions. The benefit that we can take from the student project are: the student project is important for biological science for medical students, especially in molecular biology. And this topic is important for medical development, for updating patient treatment.
My name is Putu Anindya Dhamma Pradevi. The last three digits of student number is 159. And I will explain the activators and repressor of transcription. Transcription factor is an element that binds to promoter proximal regulatory element and enhancer in eukaryotic DNA which stimulate or repress transcription. Transcription activators and repressors are modular protein containing a single DNA binding domain and one or few activation domains for activators or repression domains for repressors. Transcriptional activator is a modular protein composed of distinct functional domains that increase transcription of a gene or a set of genes. The functions of activators are to promote gene transcription and in some cases are required for the transcription genes to occur, which is why they are considered to have positive control over gene expression. They have two main domains. The first one is DNA binding domain that binds to a DNA sequence specific to the activators. And the second one is domains that function to increase gene transcription by interacting with the other molecules. Is a DNA or RNA binding protein that inhibits the expression of one or more genes by binding to the operator or associated silencers. DNA binding repressor function by blocking the attachment of RNA polymerase to the promoter, thus preventing transcription of the genes into messenger RNA. RNA binding repressors bind to the mRNA and prevents translation of the mRNA into protein. This blocking or reduction of expression is called repression, and they have two functional domains. The first one is DNA binding domains, which is function by interacting with other proteins, and the second one is repression domains that is continued function when fused to another type of DNA binding domains.
I am Gusti Agung Krisania Mahaputri with the last three digits of my student number are 143. We'll explain about molecular mechanism of transcription, activation, and repression. The DNA in an eukaryotic cell contains two types of chromatin. Euchromatin that actively transcribes it, and heterochromatin the more condensed and silent one. There are two general mechanisms of activators and repressors to bring their impact into transcription process. First, indirect effect by modulating the chromatin structure. Second, direct effect by interacting with Putu and general transcription factors. The chromatin structure can be actively transcribed when it's in euchromatin form, so that the structure must be modulated. Chromatin structure can be modulated by the interaction of protein with hypoacetylate and terminal tails of histone deacetylase complexes, histone acetylase complexes, and dissociation by SWI or S and F chromatin remodeling factors. Besides mediator, which is a molecular bridge between activation domain and RNA polymerase to help direct effect process. Activators can bind to a distant enhancer to interact with transcription factors that bind to a promoter. Specific site of activators also produced by cell in order to express this particular gene in transcription process. That's all. Thank you.
My name is Putu Nadia Anandita Prasanthi and my last digit number is 150. Let me explain about the regulation of transcription factor activity. The activities of many transcription factors are indirectly regulated by binding of extracellular proteins and peptides to cell surface receptors. These receptors activate intracellular signal transduction pathway that regulates specific transcription factors. Nuclear receptors constitute a superfamily of dimeric carbon-4 zinc finger transcription factors that bind lipid-soluble hormones and interact with specific response elements in DNA. Hormone binding to nuclear receptors induce conformational change that modify their interaction with other proteins. Heterodimeric nuclear receptors, like retinoids and vitamin D, are found only in the nucleus. In the absence of this hormone, they repress transcription of target genes with the corresponding response element. When bound to their ligand, they activate transcription. Steroid hormone receptors are homodimeric nuclear receptors. In the absence of hormone, they are trapped in the cytoplasm by inhibitor proteins. When bound to their ligand, they translocate to the nucleus and activate transcription of target genes.
My name is Prabha Dewi and my last 3 digits are 146. Different mechanisms of transcription termination are employed by each of the eukaryotic nuclear RNA polymerase. Transcription of most protein-coding genes is not terminated until an RNA sequence is synthesized that specifies a site of cleavage and polyadenylation. Transcription of the HIV genome by RNA polymerase II is regulated by an anti-termination mechanisms that requires cooperative binding by the virus-encoded TAT protein and cyclin T to the TAR sequence near the 5' end of the HIV RNA. During transcription of Drosophila Hitchcock genes, RNA polymerase II passes within the downstream promoter proximal region, which interruption in transcription is released when the HSTV transcription factor is activated, resulting in a very rapid transcription of the Hitchcock genes in response to the accumulation of the nature proteins.
My name is Putu Cahya Prameswari, with the last 3 digits of my student's number are 151. Gene promoters are the sequences of DNA which is located in the early end of the area that give codes to the genes and also contain several cis-acting elements. The cis-acting elements is a region which function is to bind specific protein that involved in the transcription process. In most transcriptional units, the promoter is located near the start site of transcription but is not transcribed by itself.
I'm Philbert Purbowo, and the last 3 digits of my name is 148. Gene promoters play a very important role in transcription, as it is the region gene transcription is achieved. Without it, the process itself won't start. It instructs the direction of transcription, how to read the DNA strand, and also identifies the same strand to be read. It contains of 3 elements, which are maximal and distal promoters. Each play a different role. Core promoters play a role as the minimal part required for the initiation to happen. Proximal promoters play a role as the binding sequence, while distal promoters also play a similar role to proximal promoters, however with a weaker influence. Thank you.
My name is Putu Anindya Dhamma Pradevi and the last 3 digit of my student number is 159 and I will explain the types of gene promoters. Initial general conclusion are drawn when promoter patterns are generated. They exhibit similar pattern despite this promoter sequence being less conserved between each species. A specific form for each promoter sequence is found in promoter patterns, which is formed by vertical position of this cluster. From these patterns, researchers classify promoters into 10 general types of promoters. The first one is CG-based promoters. The characteristic is represented by patterns containing a high percentage of C plus G and high kappa IC values, and the right side of the patterns is predominant while the left side is significantly less pronounced. These patterns of this class are particularly characteristic for genes from Homo sapiens. The next one is ATCG balanced promoters. Their sequence show an almost balanced G plus C and A plus T content. The right and the left sides of the patterns tend to share a relative 2-fold rotational symmetry. These patterns are characteristic for gene promoters in Homo sapiens and Oryza sativa. The next one is base promoters. Representative patterns are distinguished by high A plus D% and kappa IC values. The left side of the patterns is predominant while the right side is significantly less pronounced, and is a characteristic for genes promoter from Drosophila melanogaster and Arabidopsis and are less common in humans. The next one is ATCG compact promoters. The patterns characterize promoters with centrally disposed clusters leading to the formation of a round-shaped patterns, and is a characteristic for gene promoters from Arabidopsis thaliana. The next one is ATCG middle promoters. The characteristics: the patterns are characterized mainly by promoter segments containing A plus D and C plus G balance value and higher than average kappa IC values. The right side and the left side of the patterns are equally distributed. The next one is ATCG less promoters, and the characteristics are represented by an abrupt transition between two C plus G threshold levels. The right side and the left sides of the patterns is equally distributed, but however, some segments around the central regions are missing or have a lower density. The next one is AT less promoters. The characteristic is the sequence belonging to these promoters exhibit a high frequency of short CG-rich sequence. Although both sides of the pattern show relative to fold rotational symmetry, the cluster from the left side of the pattern exhibit a lower density than those on the right. AT-less patterns are generally rare and found equally in all four species, but are slightly more frequent in Homo sapiens. The next one is CG spike promoters. It is represented by a long, repetitive sequence with a high content of C or G nucleotides. And this pattern exhibits a central part and an elongated right side containing small density cluster. These patterns are common in Oryza sativa and common Homo sapiens. The next one is CG-less promoters. They are distinguished by a high frequency of short AT-rich sequence and are more common in Oryza sativa and Arabidopsis thaliana. The left and the right side of the pattern tend to be equally distributed. However, the cluster from the right side of the patterns exhibit a lower density than those on the left. And the last one is AT spike promoters. They are represented by a long repetitive sequence with a high content of A or T nucleotides. These patterns exhibit a central part and an elongated left side containing small density clusters, and they are found especially in human in tissues that require high levels of gene expressions.
My name is Jacinda Isadora Mannuela with last 3 digits of my student number 147. Promoters play an important role in transcription or the process of copying DNA to create a blueprint for making proteins. Promoters contain DNA sequence that can help RNA polymerase attach to DNA. The presence of a promoter is a sign of the direction of transcription from upstream to downstream of the gene. For avoiding misdirection in the gene transcription process, the promoter also acts as a binding site for enzymes used for the formation of messenger mRNA. After binding, RNA polymerase undergoes the addition of a phosphate group, which causes the initiation complex to become active and the gene transcription process begins. Inhibitors, a substance that can decrease the rate of or stop a chemical reaction by blocking the active site. Inhibitors can bind to receptor protein so they can bind to DNA. It's called co-receptors. They also can bind to activator protein so they do not bind to DNA. For the examples, inhibitor function is to inhibit the process of cell formation in cancer patients, and another example is DNA methylation.
Now, before we look into its mechanism, it's important to know that transcription factors are proteins that regulate gene expression in eukaryotes by binding to a specific region of the DNA, which is the promoter region of the gene. Their duty is to make sure that a gene is expressed at the right cell, at the right time, and at the right intensity. The binding of a transcription factor to a promoter region results in either one of the two responses: increase or decrease the transcription rate of the gene. The mechanism to stimulate transcription is as follows. After the binding of a transcription factor to the promoter region of the gene transcription can begin with the binding of RNA polymerase to the aforementioned gene, and transcription will begin. On the other hand, to inhibit transcription, a repressor protein will bind to the transcription factor. Thus, the transcription factor cannot bind to the promoter region of the gene, and transcription will not happen.
Hello, my name is Nadia and Sembiring, and my last 3 digits of studio number is 155. The promoter gene is a region of DNA where specific proteins will bind to each other and start the transcription process. Promoter genes play a critical role in gene expression. One mistake in the transcription process can produce different proteins. This can harm the balance of the genetic order in the body. The presence of mutations in this promoter gene can initiate the emergence of diseases in the body, for example, cancer. Cancer is a condition in which cells in the body grow abnormally and uncontrollably, destroying body tissues. Bladder cancer is one type of cancer resulting from mutations in the promoter gene, with an estimated death rate of 200,000 people per year. The study concluded that mutations in the core promoter gene could lead to the expression of the faulty gene, which can trigger the development of cancer cells and cause bladder cancer.
My name is Skylenn Hertanto Tan with the last three digits of my student number 145. So what is the conclusion? Firstly, we know that transcription is a process of converting the DNA into RNA. The gene promoters that the sequence of DNA which is located in the early end of the area that give code to the genes and also contains several cis-actin elements. The cis-actin elements is a region which function is to bind specific protein that involved into the transcription process. The messenger RNA serves to convey the message of the transcription, and the promoter contains DNA sequence that can help RNA polymerase attach to the DNA. The promoter then tells the polymerized RNA to enter the data box in the gene, namely the thymine and adenine-based repeat complex. It eventually forms an initiation complex at the promoter and binds to the DNA transcription initiation factors. After binding, the RNA polymerase undergoes the addition of a phosphate group, which causes the initiation complex to become active and the gene transcription process begins. And if there's an error or abnormality in the promoter gene, this can cause negative impact in the body, because one mistake in the entire genetic coding process can cause different proteins to be produced which can initiate disease in the body. For example, is cancer.
My name is Kinan Kalisha with the last three digits of my student ID number are 152. So, from this student project, we know that transcription is a crucial process. Small changes of the base sequence during that process can cause error in the manufacture of proteins, which lead us to mutation. All we have to do is prevent the cause of mutation with a healthy lifestyle, because all of the factors are under our control. So, let's try to avoid smoking and being near smoke, wear sunscreen to minimize the excess of sun exposure, and limit consumption of processed meat and grilled food. Thank you.